So it's a pleasure to welcome all of you for this first webinar, which will be the first of a series of webinars the Global Virus Network is now organizing. I'm Christian Brichot. I'm a professor in medicine at the University of South Florida and president of the Global Virus Network. And before coming to the US, I have also been president of the French NIH and president of the Institut Pasteur. So the Global Virus Network, briefly, is really a network of scientists, of individual scientists. This is science-driven. This is independent of governments, very much working in collaboration and with complementary activities to the national and international institutions and agencies. So again, the Global Virus Network, this is really the opportunity to merge all the best experts in virology all over the world to really focus on the fight against viral infections. So today it's my great pleasure to introduce Linfa Wang from Singapore. Linfa has been for now several years at the forefront of the activities of GVN with obviously its intrinsic highly recognized scientific activities. But in addition to this, his commitment to work with GVN, for GVN, and I really want to thank him for this. Actually, we were planning with Linfa and Sharon Lewin to organize early May the first regional Southeast Asia GVN. And unfortunately, we had obviously to postpone due to COVID-19, but this will come back. And this is just to illustrate the commitment of Linfa for our network. So having said that, Linfa, again, thank you very much. And we look forward to your seminar. Please.
Thank you, Christian, and thank you all the team for GVN. And I think Christian's right. I'm very dedicated, to promote the global viral network in Asian area. And we, as he said, we had a GVN meeting organized with PDI and the Peter Doherty Institute in Melbourne, and Duke and Yes Medical School in Singapore. So we were going to host a GVN Asia meeting on May 3rd this year. So, if without the lockdown, there would be a fantastic meeting. We would obviously focus on COVID-19, but because of the lockdown, we could not go ahead. So I'm very pleased to present the first of many in this GVN series of COVID-19 related talks. All right, so what I'm going to talk something is that usually people don't take it seriously, the serology. In an emerging disease outbreak and pandemics, you have vaccine, you have drugs, countermeasures much more sexy than a test of serology. But I have been really, working in this emerging infectious disease, especially zoonotic diseases, for the last 25 years. And personally, is a strong believer that serology has an important role to play. And for COVID-19, I think it is. So I titled this The Role of Serology in the Next Phase of COVID-19 Investigation and Response. But I thought just to start with, the other people in the audience are very familiar with this already. So I'll be very, very simple to go through the outbreak and the disease of the virology. And then, unbiased, I'm going to list 3 current and future challenges, and all of them, of course, relate to serology. So then really the bulk of this is to talk about how serology can play a role in response and facing the challenges. So just one slide on the outbreak. By now, we all know it's COVID-19. Start with the 2019 novel coronavirus outbreak. And it started in December 2019 in Wuhan, China. And as of 2 days ago, that's the WHO number, has resulted more than 6 million infections and close to 380,000 deaths. As I said, I have been working in this field for the last 25 years. I will show you later, start with hantavirus in 94 in Australia. I think that this shocked me into really, COVID-19 still shocked me into really aspects. One is that I was actually in Wuhan, January 14th to 18th. So that was actually a very critical week before the authority declared human-to-human transmission and before the shutdown of the Wuhan city on January 23rd. But honestly speaking, as an expert in this field, I never realized it's going to be that big and that serious. So that's, I think, one shock to me. The second shock to me is that if you want to do a hot map, you know, so sort of the zoning of the world, you will never predict, right, the most affected area, you know, the dark area, especially for the United States and also Europe, you know, is heavily affected. So these are the two really unexpected for me, you know. In terms of another outbreak caused by backbone virus, that's not a surprise to me. What's surprise is the scale and also the most heavily affected epicenters now come to the most developed nations in the world. So this is the, you know, just very quickly run through, right? December 8th now is the first really confirmed COVID-19 case, although that's diagnosed retrospectively. And really late December, you start have this severe pneumonia of unknown etiology. And by January 7th, that the Chinese CDC basically said that not 100% sure, but we believe that it's a novel coronavirus. And then the first death was recorded in China, January 11th. January 13th, that first confirmed cases outside China in Thailand. But I have talking to the scientists over there. Actually, they had a PCR results a few days before. So the export of the first case was actually early January. And then WHO declares a global health emergency. And so I was one of the 16 members on that committee. And by February 11th, you know, the ICTV really renamed the virus as SARS-CoV-2, and WHO changed the name to COVID-19. And then another month later, WHO declares pandemic. So these are very important, you know, WHO's milestones. But as a scientist, you know, working in the field, I think that there are two really important, you know, milestones or maybe dividing sort of points. One is January 12th. It's the first complete genome sequence become publicly available, you know, by WHO. And of course, it's from China to WHO, but at least publicly we can assess that. In this day and age, in terms of pandemic response, you know, you all know how important it is to have a genome sequence. The second important milestone is January 20th. That's the date, you know, Chinese authority declared that there is evidence now human-to-human transmission. 2 days ago, I was in Wuhan, and the official still says there was no human-to-human transmission. So this is a big dividing sort of a point because the psychology and the society response is really different. And 3 days later, as we all know, the whole city, 11 million, Wuhan was shut down. Okay, so very early days, you know, the Chinese authorities spent a long time really trying to say it's not SARS. And so the question is COVID-19 SARS or not SARS? Depends on what you want to focus. If you focus on the disease, then SARS is severe acute respiratory syndrome. For COVID-19, now we know more and more actually, majority of patients present with mild disease or even asymptomatic. So it is not SARS, right? But if you look at the virus is 80% genetically identical basically to SARS coronavirus. So the SARS-CoV-2 is 80% identical. So in virus taxonomy, different families, you know, we really group them into family, genus, and species. And for coronavirus, there is a certain way of grouping virus into species. So in this case, that there is a species group called the SARS-related coronavirus, and both SARS-CoV-1 and SARS-CoV-2 are in this species. So if you look at the virology, then it's the same virus basically, different strands, right? So the analogy is that, you know, all human beings are one species and we have different sort of ethnic, you know, ethnicity. So in virus, we call different strands. So that's how close they are. And the naming of a virus is complicated, and I will show you later that it's never simple. It's not a purely scientific exercise. Nowadays, we have to, you know, really consider the diplomatic area, the politics, everything. So unfortunately, naming of a new virus is not a simple matter anymore. So just really very briefly to introduce the virology. So on the left, that's the sort of computer enhanced imagery of a coronavirus, and the red really paths indicates this important protein that projected from the surface, and it's called a spike protein. And the spike protein is the reason that if you do a section that the virus looks like a crown, and hence the name of coronavirus. And spike protein is also very important structure protein because it's important for docking onto the virus receptor to mediate entry, and also important for membrane fusion with the host. And the genome of coronavirus I will touch later is also unique. It's the the longest RNA genome in any viruses. So the spike protein, is important. It obviously has many domains. The N-terminus, the half, almost there's a cleavage site by protease, and then you form the so-called S1 and then the S2 on the C-terminus. The S1 is responsible for receptor binding, and S2 is responsible for membrane fusion. And in the S1, then you have the even more important called the receptor binding domain, that green box that I'll come back later. And in the 3D structure, here's the membrane and of the virus, and the RBD is really on the tip of the spike surface because that's the point that they need to contact with the virus. And in terms of virology, I already said that coronavirus is unique because RNA virus are inherently less stable because their polymerase has a low fidelity. So during replication, RNA polymerase are prone to mutations. So for that reason, the RNA virus genome usually is small. And if you want to have a large coding capacity, then the virus has evolved to a segmented RNA virus, like the influenza virus have 8 genome segments. So coronavirus is unique. It has 30 kilobase, but it has a single genome, no segmentation. The reason is the polymerase has a high fidelity. And also that, it's a still mystery that SARS-CoV-2 seems to be genetically more stable than SARS-CoV-1 or the MERS coronavirus. And that's, first reported by WHO, and now I think many more papers. This now has been considered as one sort of a possibility that actually the SARS-CoV-2 virus has been circulating in the human population maybe longer than just December 2019. Because they already seems to be stable and adapted to human population, maybe initially less kind of pathogenic and undetected. The important discovery, now everything moves really fast. As soon as the virus was isolated, many groups, including the Wuhan Institute of Virology, proved that just like the SARS-CoV virus, it used the ACE2 as a receptor, but with a much higher affinity. So now this has been at least explained as one of the reasons that the virus transmits among the human population more efficiently than the SARS coronavirus. The most related virus is a bat virus, 96% overall genome identity. So that's from bats in Yunnan province in China, which was sampled 2013. But several groups have published papers now. We identify coronavirus from pangolin, has overall genetic sequence is lower, around 90%, but the receptor binding domain is even more related than the human COVID-19 virus than the bat virus. Okay, so personally, I have been working in this field for exactly 5 years. In 1994, when I was in Australia Animal Health Laboratory, we discussed this virus called Hendra virus. So this is the first modern bat-borne virus spill over from bats and goes through horses and into human. 5 years later, in Malaysia and Singapore, where I am now, there's a close related virus. Hendra and Nipah should be in the same species as well. That jumped out of bats and goes through pigs and to human. And we all know SARS-CoV-2 to 223. We now believe that civet is the intermediate host. And MERS is slightly different. I think the jump from bats to camel may happen many years ago before we detected camel to human spillover. And 2014, the Ebola West Africa, Ebola has been with us. We know Ebola for the last 40 years, but the largest outbreak was 2014. And this one, we believe that does not involve intermediate host directly back to human transmission. And in COVID-19, I think it's, although it's not 100% conclusive, but most people believe most likely the virus origin from bat. The question is, what's intermediate host, or does it involve intermediate host? So currently the front runner, but I have to say it's not conclusive. the front runner, everybody really get excited, is a virus from pangolin, as I said, So from this phylogeny tree, as you can see, this is the cluster of this COVID-19, virus here. And the red dot are the pangolin viruses that just been discovered recently, early this year. And the bat virus, when I say 96% identical, is here. So the bat virus is still much more close related than the human virus than the pangolin virus, but the pangolin uses the ACE receptor very, very efficiently. So the famous, seafood, Huanan seafood wholesale market in Wuhan, that was believed to really middle to late December, the cluster cases over 40, and the 70 to 80% of these cases all had a connection with the market. So this is very similar to the outbreak of SARS in 2002 and 2003 was also linked to live animal market. But this is the menu, if you like. I apologize, it's in Chinese. And this is the menu of this market. And, my friend in Wuhan took it and sent to me. They sold almost everything you can think of, but no pangolin, So is it because they never sell pangolin, or because it's illegal to sell pangolin? That's why they don't have this in the open sort of menu. But civets, civets was sold on the market, and we know civets is susceptible to SARS coronavirus, and it's susceptible to SARS coronavirus 2, okay? So I think the question of this is still unknown. Challenges, I'm going to present 3 different areas of challenges. Challenge 1, I'm going to contrast this with SARS, right? So SARS started in November, 2002. So for some reason, in China, this virus always start in later November, December, and it peaks around March, April. But with the international really effort, and the WHO really called it a very good international effort, 6 months later, really the whole pandemic was under control. And so it was 8,000 cases with 774 deaths. And so basically was eradicated or contained by July 2003. COVID-19, 6 months into it now, as I already said, over 6 million infections, 380,000 deaths. And I think, let's be honest, right? The containment or eradication of COVID-19 is almost impossible with everything's going on in this world right now. So the challenge is that, we can lock down easily, we can, but unlock is difficult. The challenge is every country and the whole world facing is the exit strategy. How can we exit this, unusual lockdown and most of the country is doing? Challenge number 2 is that without any doubt, this exit strategy, vaccine will be a very important part. Without an effective vaccine, I just cannot see how we can effective exit strategy. So, but we're still a long way away from a licensed human vaccine. And also the correlates of protection is poorly understood right now. And longevity of protect immunity will not be known until, many years later, at least. But I think that if, again, SARS coronavirus 1 and 2 are the same viral species. So in the last few months, from January, we have been sampling SARS survivors. Singapore was one of the worst hit country in Southeast Asia. So we have many sort of SARS patients survived the SARS 17 years ago. And so we had samples that took in 2003 and 10 of them, and we have more samples taken this year. So we just published this paper, and the original really aim of the study is to look at cross neutralization by SARS patient sero towards SARS coronavirus 2, in the aim to say if they really cross neutralize, because initially the German and the Chinese paper said they can cross neutralize, then we were thinking that maybe we can ask these SARS survivors, and there are more than 200 in Singapore, and could they be the donor to donate a convalescent plasma, and we can use as a first line of defense as a therapy. But, to our disappointment, so the data is here. So S is the SARS patients from S1 all the way to S12. And sampled in 2003, we say it's less than 1 year convalescent. And we have 9 years, 14 years, and 17 years. When we wrote this paper, we had less patients, now we have more. As you can see, they neutralize SARS coronavirus very well. And to our surprise, even 17 years later, you look at that, some still have very high titers, but they don't cross-neutralize with SARS-CoV-2, okay? So on one hand, we were disappointed there was no cross-neutralization, but on the other hand, we discovered something really exciting 17 years later, that we still have these survivors carry neutralizing antibody. Of course, you know, when you say these two viruses are same species, but we don't know if COVID-19 will behave the same, but at least this presents some hope to say, if a patient have survived this COVID-19 and recover, become convalescent, if they have neutralizing antibodies, hopefully they will last many years to come. Challenge number 3 is the role of animals. You know, this is my kind of expertise area. You know, I work at the animal-human interface, and I was the one led the team to discover that, you know, SARS came from bats. So, but currently, the role of animals in the current really outbreak remain elusive, as I said. We even don't know, you know, what's the true natural reservoir host, and not clear whether intermediate animal host was involved. Most people will believe, you know, there was an intermediate host, because bats usually carry virus at very low levels. So you need an intermediate host, which is also a naive host. They may or may not display disease, but usually they replicate the virus to very high titers that facilitate animal to human transmission. So if so, which animal? As I said, you know, pangolin is the front runner, but I don't think that that's it. We need to really continue to search. And the search should not be limited in China, because we know there's lots of animal trading, you know, into China, some legal, some illegal. So there is a possibility that the true reservoir or the intermediate host actually come from outside. And then for COVID-19, that's a new challenge and a concern, is what we have observed is this event called a spillback or reverse zoonotic transmission. Basically, we have evidence now infected humans can give the virus to animals. This includes dogs, cats, tigers in a zoo, and minks in a mink farm. You know, so this is serious, because if these 4 animals are susceptible and can, you know, be infected by reverse zoonotic transmission, there'll be more animals. And the worst, you know, you can get is this reverse transmission, establish a new natural reservoir host. You know, that will be very difficult. So now let's come to this role of serology. You know, what we have the challenge is also that, you know, serology can help. Is a herd immunity, exit strategy very important. Some countries are seriously consider this individually. Let's, you know, can we give them immunity passport? The longevity of the immunity, convalescent plasma therapy. Not all infected patients after they become convalescent will have high titer neutralizing antibody. So we need to know this. Vaccine, you know, first in the clinical trial, but especially after the licensing and for human use, once we have a mass vaccination at a population level and individual level, we all want to know whether the protective immunity has been reached. And then search for this animal and also the reverse zoonosis. So for the first 5 challenges, basically what we need to measure is functional neutralizing antibodies. The second, the last 2, the blue dots, is we want a serology that can do species independent testing, because you know, like in Australia for the hantavirus, we examined 42 different wildlife, including kangaroos and koalas. So I think to develop a specific test for each animal is going to take long time and very hard to have positive control. So what we need is a species independent serology for animal studies. So for antibody response, let's come to this genome figure again. So, you know, a 30 kilo base sort of genome, the protein is around 10,000 amino acids. So there's many proteins and many epitopes. So when you have a virus infection, basically, you generate 2 types of antibody. One is just a binding antibody, which binds to the virus or different virus protein, but they don't neutralize the virus. The functional antibodies and neutralizing antibodies, in most of our infection, that's less than 5%. And in coronavirus, because the large genome, it's less than 3%. So the neutralizing antibody is really mostly, not exclusively, target to this spike protein, the grain protein, and to this receptor binding domain. Okay, so that's important. I'll come back later. Okay, so the gold standard to detect virus neutralization antibody is this live virus neutralization test. So here's the diagram, virus will bind the receptor. In this case, it's ACE2, and then there's a cleavage of S1, S2, and then the membrane fusion starts and the virus goes in. In the presence of neutralizing antibody, there's many steps you can block, but most important step is the receptor and virus interaction. So the RBD and the ACE2 interaction. So this is the gold standard because it's the most specific assay you can have for any virus, and the COVID-19 the same. The disadvantage is for COVID-19 is a BSS3 pathogen, so you need a BSS3 lab. Even in the most developed nations, this is a very limited supply. And it's tedious and slow. It takes 3 to 5 days to do the neutralization. And you need a highly skilled staff like that. The training usually takes months before you can go into it. And the read out is the cytopathic effect. Basically, you see if the cell has been killed or not. So you need a very experienced eyes, like, on the left, there's no neutralizing antibody, and on the right, there's neutralizing antibody. Okay, so it's very difficult. So what I did with my group is that in early March, I already see this will be a challenging area. So we were thinking, can we invent something to really address these two challenges? We want a neutralization test that can match neutralizing antibodies in a species-independent way and does not rely on live virus in BSS3. So we invented this test called a surrogate virus neutralization test. So the principle is very, very simple. So this now is a diagram saying, your virus binds to a receptor, the S2, and you get an entry. In the presence of neutralizing antibodies, the antibody will bind to the spike protein, especially RBD, and mask that site, and you cannot bind to receptor. And so the surrogate virus neutralization is basically, instead of use live virus and cell, we use protein engineering. So we transfer the ACE2 become a non-membrane protein, and we purify this and coat onto ELISA plate. For the spike protein, we chop off the most of the protein, just express that RBD domain, and we chemically conjugate host radical properties. Well, this is the enzymatic reaction we need to see the signal. So when you have this HRP RBD binds to ACE2 and you put a substrate, you see color. In the presence of neutralizing antibody, you block that reaction, and either you eliminate color or you reduce color. So you achieve the neutralization, which is expressed as an inhibition of the color reaction. So, it's easy to get an idea, but usually putting into practice is more difficult. But for us, maybe we're lucky. So within a week, we got the data. So what we did is we used the SARS-CoV-2, 3 proteins. The nucleocapsid protein, which is internal, should not make a contact with ACE2. That's our negative control. So this is a nanomolar. As you can see, increased concentration, basically it's negative. The S1 protein, which is the half of the spike, and as you can see, that it binds in a dose-dependent manner, but not as good as RBD. RBD is fully exposed, and it binds to ACE2 the best. Okay, so that's very encouraging. Now the next question is, can we use the human sero to inhibit that? So we only use patient 1 and patient 2 and the healthy control 1 and 2. As you can see, the red and blue has a dose dependent. At low concentration, they have an inhibition, and when you dilute out, inhibition goes out. So we got really excited because if it works for COVID-19, it should work for SARS as well because they use the same receptor. So we immediately, expressed that RBD of SARS coronavirus and did the exact same thing, HRP conjugation. And, in Singapore, we have SARS patient sero. So we just did the 2 sero and that beautiful data, just like the COVID-19. So, with that, we immediately patent this and we get a company to commercialize and develop this into a wide variable test. But the second challenge is to say, now this is a surrogate neutralization test. And one of the beauty of neutralization test is not only is specific, but it's species independent because we don't need a secondary antibody like ELISA or other tests, So this is the data. So what we did is for COVID-19, we don't have an infected animal yet. So we use immunized animal, the mouse and the rabbit. So this is the naive, negative control, there's no inhibition. And as you say, the 3 rabbit sero, the red, the blue, the green, and the orange, very good inhibition. And the mouse sero, the other 3 also have very good inhibition. Same for SARS. For SARS, I have some infected sera when I was in Australian Animal Health Laboratory. These are the ferrets infected with live SARS virus. And these are the rabbits also immunized with the whole virus, not infection, but at least the whole virus. As you can see, all very beautiful, very sensitive, and very, very specific neutralization. So this is great because, as I said, we already know from the reverse zoonotic transmission, cats, dogs, tigers, minks have already got infection from human, and I said more animals most likely. Naturally, we know pets, pangolins, and civets can be infected by this virus. So now we have a test basically without any modification you can use in hospital, in veterinary clinic, or you can use in the jungle and looking for wildlife animals. The second question people always ask is the surrogate neutralization. We know the one gold standard for why it's gold standard is very specific. It does not cross-react with cross-related virus. So what we did is here is we checked with the MERS virus sera, okay? 30% is our cutoff for the neutralization inhibition, nothing. 229E and NL63, these are commensal human coronavirus, nothing. OC43 is another commensal human coronavirus, nothing. SARS coronavirus is interesting. In the 2013 or 2003, 17 years ago, there's no cross-reactivity, but 17 years later, we see low cross-reactivity. Remember, in the virus neutralization, we did not see cross-neutralization. So this already indicates that it's more sensitive than virus cross-neutralization, which makes sense. This is a protein-protein interaction, and we detect it biochemically, so it's very sensitive. But the beauty is now we have two surrogate virus neutralization. We have the coronavirus 2 and the coronavirus 1, right? So if you have any doubt to say, especially in a country like China, Singapore, which was affected by SARS 17 years ago, if you detect a low cross-neutralization in a healthy or in a surveillance healthcare workers, all you have to do is do these two surrogated virus neutralizations side by side. As you can see, these are the SARS, in 2003. Clearly, SARS, the surrogate neutralization is much more sensitive at an inhibition of 70 to 90%, whereas the COVID-19, the coronavirus 2 surrogate neutralization is only below the cutoff. For this 17 years later, somehow that the homologous neutralization, as you can see, reduces. So in 17 years ago, for SARS, neutralization is here. Here now we have a wide spread. Some are still high, but others are very low. But for the cross neutralization, for example, this 2 to 3 now is about 30%. This is a very interesting observation. We don't know the mechanism, and I can tell you that the N protein actually go the other way around. They have a high cross-reactivity 17 years ago, but 17 years later, the N protein antibody is gone, but the RBD antibody is still there. So this is interesting, but it's a different topic. I don't want to go into too much. Okay, so the other challenge people ask us is it's a surrogate virus neutralization. So you have to correlate with live virus neutralization. Currently, there are two different types of live virus. One is using the COVID-19 virus and has to be done in BSS3. And as you can see, this is the correlation. We have 60 series, especially when you have high sera, high neutralization titers, correlation is beautiful. When you go to lower end, then as you can see here, these are the live virus neutralization negative, but the surrogate virus neutralization is still weakly positive. The other test now, a lot of people are using is the so-called pseudovirus neutralization test. So you use a VSV or lentivirus and you pseudotype with the spike protein from COVID-19. So we have this as well. So it's a pseudovirus. The advantage is you can use in BSA2. The disadvantage is you still have to use live cell and you still have to use live virus, cell and viruses, right? But the correlation now is even better, as you can see at the low end. So because this one now is we are really measuring only the S and RBD binding neutralizing antibodies, whereas for the live virus, you may be measuring other antibodies that bind into different parts of the virus, but they neutralize, okay? So this is very, very encouraging. And currently we are create a panel of standard. We will calibrate among these 3 different virus neutralization, and hopefully that's will be the beginning of establishing what we call is a international units for COVID-19 neutralization. That is highly needed, I think. Just like rabies virus, if you do a vaccination, the doctor can send your serum away and you come back with international units. So we need something like that for COVID-19, and we are working toward that. In the validation is that we have 2 cohorts, that one in Singapore, and now we have increased number to 170 positive and the 200 negative. In China is 50 positive and 200 negative. And as you can see, it's incredibly good. The specificity is 100%, sensitivity is around 98% in both cohorts. But of course, we need to be careful because we still have zeros that PCR positive, but they don't produce enough neutralizing antibody. And I think that's like every virus infection. Some people get virus cleared and become convalescent without inducing enough neutralizing antibodies. So maybe the T cell immunity play a more important role in these patients. So we have to be careful that the sensitivity to serology is only as good as the patient's seroconversion. Okay, now, so this is the question everybody's asking, and WHO is hesitating really try to make any conclusive remarks. Can antibody testing predict protect immunity? To answer this question, we have 2 really kind of unknowns, or not unknowns, really challenges. The first challenge is that every commercial test on the market, whether it's a rapid test or ELISA test or a high throughput test, only measures binding antibody. So this is the question. Does binding antibody equal neutralizing antibodies? Obviously no, and I'm going to show you data. So that's why it's important to have a functional neutralizing antibody test rather than just an antibody binding test. The second is more difficult. Even if you have high level neutralizing antibody, does it equal to protect immunity? Now, from past experience, you would say in most viral infections, in most cases, you can say that's correct. Okay, so there is still being debated, but even Tony Fauci basically says that. So this is like he just a press conference just a few days ago. People asked him the question about this data from the Moderna sort of vaccine trial. He said the thing that made everybody so cautious optimistic is that we did not just see binding antibodies. It was clear that neutralizing antibody was produced. And at the level you would predict be protective, and then he qualified this if in fact neutralizing antibody is going to be correlates of immunity, and he add on, which is a reasonable assumption, So Peter Doughty in Melbourne also made that to say if you have really high neutralizing antibody, then I think most likely you'll be protective. My personal take is that to say programmatically, this is the only indicator we can conveniently measure. And also I believe with these 2 top scientists that it's a reasonable assumption that if you have high neutralizing antibodies, then most likely you'll be protected. Okay, so since we submit a preprint of this SVNT, we have much more data in the revised paper. So what we did now is in collaboration with GeneScript, we produce antibodies, monoclonal antibodies, by immunize mouse and rabbits, llama, and also using convalescent humans that we have generate monoclonal antibody from 4 different species, and we screening by IBD binding. Then we test in this SVNT. So the first 4 is mouse monoclonals. As you can see, the binding is beautiful to IBD. Look at the red one, is on the top. But if you do the testing in our SVNT test for neutralization, then you can see the inhibition actually does not work for the top red and the orange, only works for the blue and the green. So this is beautiful. The SVNT is measuring truly neutralizing antibodies, whereas our ELISA is measuring the total binding antibody. So that's what we want, and we now demonstrate. So with the limitation of time, I'm not going to give all the data, but the human is really equally interesting. We choose the 4 monoclonal antibodies, and then the green and the orange are the good binders, and these are not as good. But when you do SVNT, for example, this orange is now fair, and the blue one actually is reasonably good, but not as good as the green, okay? What's really interesting is that when we try to do a mix, so in this case, let's say mix this blue and the green, what you get. Here's the data. If you mix the blue and the green, theoretically, if you take this and take an average, if there's no synergy of these two monoclonal, you should get this dotted line, right? Because that's the average of these two monoclonal antibodies. Instead, we get this purple solid line. So there's a huge increase actually between the dotted line and the solid line. What we're observing is something that have been observed in HIV, Ebola, ISV. It's called neutralization synergy or the neutralization epitope synergy. So you have this in polyclonal serial that happens as well. You have so many antibodies bind to different epitopes of neutralizing. Individually, they may not be a strong neutralization epitope, just like this blue one. But when you mix with the green one, they actually get a synergy and they get a better neutralization. Why this is important? Because our surrogate virus neutralization detects neutralization antibody not only in a species independent way, but also in an isotype independent way and in an epitope independent way. So that's really important. So this is my last sort of a data slide I want to show. I'm going to go the other way around. Let's focus on panel D first. So these are the 22 human convalescent serials selected from a panel of 200 in Singapore. And we have grouped them into 4 different panels classes. One is they have high IgG and high IgM. So as you can see, this is ELISA determining isotype specific RBD binding antibodies. So IgM high, IgG high, and the neutralization curve is beautiful. Not only at low dilution, it close to 100% in neutralization, but also the duration. You look at the slope of the curve, it drops slower. Okay. Now you come to this low IgM, high IgG panel C. So low IgM, high IgG, still beautiful, and the curve is still kind of goes up like that, and not very sharp drop. The third is panel A. Now we have low IgG and high IgM. They still have good inhibition, but the slope now, as you can see, goes sharper. The most interesting, exciting to me is that we have this panel that low IgM and low IgG. If you have a cutoff somewhere here, majority of them will be considered almost negative, but we know these are truly RBD binding antibody, but at a very low level. Much to our surprise is the neutralization inhibition is still at a 70 to 90% at a low dilution, which is 1 in 20. And when you dilute it out, now if you compare this panel B and the panel D, there's no comparison. That curve is much sharper, right? So there are 2 conclusions which are sort of a total surprise to us that, from this revision of the paper is that, first of all, is that, of this low IgM, low IgG, but looks like they already have synergy. In our neutralization, they have a 70 to 90% inhibition, which is pretty good. Secondly, is very clearly this is sharp. The slope is the sharpest. This is second, third, and the fourth. So what this says is IgG plays a much important role than IgM. If you look at this, high IgM, low IgG, the curve is like that, and high IgG, low IgM. I mean, maybe not surprised to the expert in the audience, IgG have high affinity and maybe contribute to more neutralization, but still very excited for us to see that when we did a very detailed titration curve in the SVNT, we reveal such information. So to finish up, the key advantage of SVNT is that, it's very specific and sensitive and safe and fast and adapted to both low throughput or high throughput. You can automate this and can be used for both human and animals. And I cannot emphasize enough, this is the only test capable of measuring neutralizing antibodies without a need of live virus or containment. Yeah, so thank you for your attention. And I have a conflict interest declaration. There's a patent on this technology and there's a commercial kit. Thank you.
Thank you very much, Linfa. Thank you very much for a great presentation and both general introductions and general points, and then very specific and stimulating data. So we can move to the questions. We have 2 mechanisms. You can just unmute and directly ask your question, which is more simple. At the same time, Linman and Kevin, I ask you to look whether some questions have been written in the parts of the screen which is devoted to conversation. But I suggest that those who want to ask questions just unmute and initiate the conversation.
Christian, we have one question at the Q&A session.
Mm-hmm.
Okay, so just read it.
Yeah, so I can read this. The question is, did you try mixing a neutralizing antibody with a non-neutralizing antibody against RBD? So I half understand the question. I think that this is maybe you want to see if there's also synergy. Is that what this is going? So the human data actually was come from there. So we mixed all 4 antibodies and we saw this purple curve. And we thought this, there's a non-neutralizing antibody become, help with the neutralization antibody. And when we dissect it further, we realize it's not. It's due to the 2 neutralizing antibody has this synergistic effect. Okay.
Thank you. So I may ask another question. I mean, now that you have this assay,
yeah.
Which is really obviously excellent.
Yeah.
Do we have access, maybe not, to some of the observations? And again, we knew from the beginning we had to be very careful, yeah, where some groups claim that they had seen either, say, a reappearance of COVID-19 in some patients or yeah, long-term persistence of the RNA based on discussion. I mean, have you been able to go back and to really solve the question as to, do we really have some cases of reinfection, or should we definitely conclude that all these cases were explained by confounding factors?
Okay, so this is a tough question, and it's a very interesting question. Of course, we have been observed this very closely, and I have a collaboration with a Chinese group. We have one patient that keeps shedding RNA for 100 days, totally healthy, and has low level of neutralizing antibodies, okay? So my personal take is that when you have 6 million people, over 6 million people infected in all different sort of genetic background, you may find a few which is really extraordinary, even if it's a reinfection or long-term shedding of RNA. So that's, I think, if it's a 1 out of a million, then even if it happens, I think we don't have to worry. What we really want to understand is one thing is clear. What we call is long-term RNA shedding. We don't call it long-term of infectious virus because live virus isolation, it's very difficult once you pass 11, 12 days. And Singapore government actually made a decision, just announced 2 weeks ago now. They don't really worry about PCR positive anymore. They really is to say, if you have a convalescent, reach the convalescent, you are healthy, and they let you go, because if the RNA CT value is high, the RNA load is low, nobody can really isolate live virus. And we don't understand why this RNA persists for so long, whether this is a subgenomic RNA or defective interferon particles. But uniformly almost that for this long-term RNA shedding patients, if you go to test their virus neutralization antibodies, their level is usually lower than the severe patient that acute very high and then stop shedding RNA, okay? So it's interesting, but I cannot give you a conclusive answer in terms of the mechanism, but definitely that looks like the neutralizing antibody now has a very good correlation with the severity of disease. Severe patients, they not only have a high neutralizing antibody, they switch on the neutralizing antibody earlier, but if these patients lucky enough to survive, they're unlikely to have long-term RNA shedding. So these are interesting observations.
Thank you so much. So thank you very much.
Yeah.
This is fascinating. We have several questions. We have a question from Damien Purcell.
Yeah.
That you can see and the
yeah, Damien, that's a good question. And in the revised paper, so Damien's question is that in the low IgG, low IgG sample, presumably is the IgA is high. No, all 9 samples have very low IgA. So now we have, when we submit the first paper, we did not have IgA test. Now we have IgA. So in the revised manuscript, we now have the IgA. No, IgA is low as well. So what I believe is that all of them, IgG, IgM, IgA, all have some neutralizing antibodies, all at low level, but synergistically, when they're together, they show this 70% inhibition. So it's a beautiful story.
Yeah, we have several very interesting questions.
Yeah.
So the next question, as you see, is from Ian Seto.
Yeah, so the mucosal IgA, okay. Yeah,
so repeat the question. So it's how important,
Yeah, how important, so this is Sito's question, how important do you think it is to also determine levels of mucosal IgA response, another correlate of protection? Okay, so this now is being debated, whether the IgA correlates better or, like what we're measuring the blood antibody, because the IgA, people argues that it's important to really provide the first line of defense. But if the virus survive and get into the systemic infection, then I think that the total sort of neutralizing antibody will be more important. But internationally, we have a collaboration in Vietnam and now in Singapore. People are looking into it to say, can we use salivas, to measure IgA or other antibodies? And so this is an interesting question, Sito, that all I can say is that maybe it's too early to say how important that part of immunity versus the neutralizing antibodies circulating in the blood. Yeah.
And then, thank you. We have a question from Konstantin Shumarov.
Yeah. So the question is, what are your thoughts about the vastly different incidence and mortality of COVID-19 in different countries and the strong dependence on per capita income? Wow, that's a tough question. Maybe Christian, you can answer that question better,
No, you are the presenter.
Yeah. Okay. The incidence rate, I think, is more interesting than mortality because, for example, the mortality or the case fatality rate, to be more accurate, like even in China, initially, we just could not understand why the case fatality is so high in Wuhan than in other cities like Shanghai, my hometown, or in Guangzhou. And then it became very clear that from my medical friends in Wuhan, it's just, the system overwhelmed, that they will not look after and they were not on the ventilator. So in China, it's very clear that the difference between Wuhan and other cities is nothing more than just the hospital care, okay? So then, your question, a strong dependence on per capita income, then I think maybe it's reflected, how early the patient can go to hospital and how well they will look after. What I'm more interested is the incidence rate. You look at our neighbors, Singapore, of course, I was personally involved. We did the incredible diagnosis and contact tracing. So other than the dormitory, in the community, we did very well. The incidence rate is low. And I can reveal that in healthcare workers, we just finished a serosurveillance. In frontline doctors, nurses, they dealt with COVID-19 patients, and out of 1,000 randomly checked, we have zero positive. So it's incredible, to me is that the good practice, but there's something else we think in Thailand, in Malaysia, in Vietnam, in Cambodia, in Laos, the incidence rate is uniformly low. So we are thinking of, is that genetics? Is that T cell epitopes cross-reacted with commensal coronavirus? Or, as we were discussing, maybe the spillover events of backbone coronavirus is much more frequent than we thought, but most of the spillover events does not lead to detectable disease. So maybe we have this virus that have induced certain sort of protect immunity, mainly T cell immunity in the population. But these are all hypotheses, but certainly very, very interesting.
Yeah, thank you. Fascinating discussion. Yeah. We have a question from Franco Bonaguro. Do you want to read it?
Yeah. Do you have any data on butcher in Singapore? That's an interesting question. First of all, Singapore, after the Nipah outbreak, we don't have live pigs anymore. We just import frozen meat. So relatively speaking, we really don't have industry, and we haven't done any sero survey there.
Thank you. So then we have a comment from Bob Gallo, which has been conveyed by Nora.
Yeah.
So I will tell it because it's a compliment for you.
Yeah, thank you.
So Dr. Gallo says that was an excellent, interesting presentation, which is true. And he has a trivial comment, but please expand more on the cat and the dog, which he thought was a rare event. And he congratulates you for the excellence of your essay, which will be a gift to the world.
Sure.
Can you comment on the cat and dog?
Yeah, thank you, and thank Bob Gallo. And yes, I mean, you can say it's rare, depends where. In Wuhan, because in the early times when you have so much human transmission going on, and then after the lockdown, the pets get very close to humans, and especially for cats, they did a survey. 15% of cats were seropositive, neutralization to COVID-19. So to me, that's not rare event anymore. That's pretty kind of high, 15. But in dogs are very rare, like in Hong Kong, they had 2, and then in Europe, there's a few, right? But then the mink farm, in Netherlands, which is pretty high as well. So I think that overall that you need to really, have very close contact with human, and the human has to be shedding the virus at high level in order for this transmission to happen. And looks like, I think the German group is going to, publish data on all the animals, and including civets and hamsters. And so whether cats are more susceptible than dog, I don't know. But, tigers in New York was infected. So maybe it is, that family is a bit most susceptible than COVID-19. Yeah.
Thank you. So then we have 2 questions actually related from Marcus Gallo, which relate to the neutralization test and assays for protection. Has this been addressed taking in account the age? And this is what I believe Marcus means with the longevity questions.
Yeah.
How is it related to age and
Yeah, so I think the first thing, Marcus, is that neutralizing antibody and the protection itself now, I think is, I guess it's very hard to establish. As I said already, in the Singapore cohort and now many other cohorts published, is that the severe disease patients have higher neutralizing antibodies than the mild disease, okay? So intuitively, it's almost the opposite. Basically says, that if you have high neutralizing antibody, your body was not doing well because the disease was kind of more severe. But if you look at the clearance of the virus as a correlate of protection, then we can say actually, if you, as I said, you have high neutralizing antibody, yes, during acute phase, you have more severe disease, but you can clear the virus faster and the RNA does not persist. Whereas in the mild patients or asymptomatic patient, their neutralizing antibody is lower and they don't have as severe disease, but some of them, their RNA can persist for a long time. So sorry, Marcus, that I cannot give a yes or no answer, but I think it's indicate that neutralizing antibody is correlated with the efficiency of clear the virus. If you define that as a correlation of protection, then I think it's happening.
Thank you. So then we have a question from Dale Godfrey.
Marcus also says to try to include the longevity question. Yeah. Then the longevity question, I think Marcus, you have to be patient. I just applied for a grant to follow the patients for the next 2 to 3 years. So hopefully I can answer this question to you later.
And Linman Lee would like to answer to this question. Linman?
Yeah.
Oh, that's just for Dr. Wang to answer.
Okay.
You can see it. Yeah.
Okay. Yeah. So the next question is from Dale Godfrey. Do you want to read it, Linman?
Yeah, so Dale has a question from Melbourne. Said, do you think there might be neutralizing antibodies that target other parts of the virus, such as other parts of Spike S2 which you would miss with your SVNT assay, definitely down. You know, this for both SARS and the MERS. For SARS and the MERS already demonstrate that, you know, the S2 monoclonal antibodies can also play a part in neutralization, maybe change the conformation. And so in our paper now, we very honestly discuss that to say that the SVNT will miss this kind of neutralizing epitopes. And but the correlation studies from the live virus and the pseudovirus would say in a polyclonal population, you know, we cannot quantify, but we are trying. At least 90–95% of neutralizing antibodies IBD targeted. Thank you.
Thank you. And then we have a question from Damien Purcell.
Oh, I could not see that. Maybe I need
Yeah,
to...
it says, does SARS-CoV-2 efficiently infect bat cells? And is there any plausible pathway for an infection of this presumed recombinant and mutated virus back into bats, confounding the understanding of the original source of this virus?
Very good question, Damien. We have a review paper, you know, maybe 20 of us in this field with bats and a bat-borne virus. We wrote a review, really strongly argued to say we have to be careful, especially with, you know, Europe and the USA right now with still so much human, and then South America, you know, and these are the continents we have lots of susceptible bats, rhinoceros bats, and so there is an opportunity for this virus to reverse spear back into bat population. We are genuinely worried, Danny. So that's why right now we are trying to mobilize the international community to at least bleed as many bats as possible of the high-risk group, and to see, you know, as Demi, you know, I'm a strong believer of serology. So if we sample the serum now, hopefully it's not too late, and keep them, and then 2 to 3 years later, if we could not control the COVID-19, and then if we suddenly see a burst of neutralizing antibody for COVID-19 in bat population, then we are in trouble. We establish a new reservoir in a different location.
Yeah, that's a very, very puzzling hypothesis. Thank you so much. Then
but it's not totally impossible, because we know these animals are highly susceptible, and we know that, you know, once you contaminate the environment, right, like this Huanan seafood market, although we could not find any positive in animals, but the cage and the walls, you swab them, it's all positive. Yeah.
So then we have a question from Franco Bonaguro.
Yeah.
Is there a cofactor including bovine coronavirus?
I don't understand that question. Is a bovine coronavirus like involved in the disease process, or what that cofactor means? Franco, can you clarify that question?
Franco, unmute and express yourself. Or type.
Yeah.
No, hello, Franco. Just unmute.
Now. Okay, so do you hear me?
Yeah.
Yes.
Okay, so one of the problem is that we have a high frequency in United States, in Australia, in England, in Germany, of the frequency of COVID-19 in the butcher factory. In some of United States, actually more than 70% of the COVID-19 patients were people working on this factory.
Okay.
And the discussion has been that probably because of the cold temperature in the factory, because people are very close, because they are not very hygiene, but we have something that is very interesting. And the province, the town with the highest frequency in Italy, is actually the area where is not the butcher factory, but is not meat production, is where there is a cow farming.
Okay.
So I wonder whether, so the vet have been studying the possibility of SARS-CoV-2 in cows, but it's not there. But it could be the reverse, that some of the bovine coronavirus instead can be interactive with the SARS-CoV-2. And what is very interesting is that the bovine coronavirus is binding the glycoprotein portion of the ACE2 receptor.
Okay.
Instead, the SARS-CoV-2 is binding the other side, is binding the protein side.
Okay.
So it could be that there is a kind of interaction between the two. So I wonder whether anyone has been studying the presence of the bovine coronavirus in humans, which is possible.
Yeah, so that's very interesting, but not to my knowledge, but I will take this down and I will talk to Linda Safe, Professor Linda Safe, Ohio State. You know, she is an expert in animal coronaviruses. Okay, thank you.
Thank you. And the last question, I believe, is from Mark Kaplan. Do you need both the RBD domain and the S protein in a vaccine to be effective?
Yeah, okay, so this is currently the vaccine is mostly use the whole spike proteins, the S protein, and some even use a trimer, but there are also, you know, just expressed RBD. The argument, you know, pros and cons, the pros and cons for the pros for S protein to say, you know, you present especially a trimeric form and you present the totality, and as Dale's question to say, you know, can other parts of the protein induce neutralizing epitopes, antibodies? Yes, even S2, you know, can contribute to neutralization. So by introduce the whole S protein, then you have maybe more authentic antigen and the more chance of hitting different neutralizing epitope, and then, you know, to result this neutralization synergy. The cons is people argue is this, you know, the elephant in the room is this antibody-dependent enhancement, ADE. We have discussed a lot. Some people say definitely it's happening in coronavirus. Other people say, no, this is only in animals and not happening in human. So some group says, just to be 100% safe, let's just focus on the RBD because this is the most efficient neutralization epitope. What you don't want is a binding antibody which is not neutralizing, which will trigger ADE. So both domains, you know, both camps, one says S protein is much more effective. The RBD camp says RBD might be safer. So, but nobody is using RBD mixed with S. I mean, that's redundant. Yeah, over.
Thank you so much. And that was a question from Hideki Hasegawa.
Yeah.
On the disease enhancement by having specific antibody.
Yeah, so that's sort of thank you, Hideki, and sort of answered that question already. You know, disease enhancing by specific antibodies, certainly in the dengue field, is still being debated, but most people believe dengue, you know, if you have a serotype 1 antibody, you can contribute to ADE of serotype 2, 3, 4. For coronavirus, there's really one animal studies, you know, you immunize the animal and then you challenge with live virus, and this animal actually have more severe disease. So then that was attributed to ADE, but I think a lot of people debate that because like the current non-human primate studies published in the last few months, you know, for COVID-19, ADE was not observed. So I think this is a still open question. Yeah.
Well, thank you so much. And we have a comment from Marcus who is congratulating you. And yeah, so really thank you so much, Linfa. That was a very great, great presentation. Thank you to everybody for joining us. This is again the first of a series from the GVN. And well, we could not dream of a better introduction to the series. So thank you so much. Have a great morning, afternoon, evening, whatever the location. And we will keep in touch and send you the program of the next webinars. Have a very good day, all of you. Thank you.
Yeah, thank you. Especially for our Australian friends, it's already midnight over there. So thank you. Okay, bye-bye, everybody. Yeah.
Bye. Bye-bye.